Sure, thank you so much, Martin, and thank you for Cintiqa for hosting this webinar today. Good morning or good afternoon to everyone, and thank you also for joining this live webinar today about the launch of our brand new preclinical imager, the Newton 7 FT, that is now integrating a 3D optical tomography module. So my name is Alexis Frances, and I'm the International Sales Manager at Vilber, specialized in the range of preclinical imaging, and I'm here with my colleague Esmeralda Narain, our in-vivo application specialist. Because not everyone knows about Vilber, I will start this webinar by giving a short introduction of the company so you can get a better picture of what we do and where we come from and how we got into small animal imaging. Then Esmeralda will go through some bioluminescence and fluorescence case studies, and I will carry on with the 3D optical tomography part and the key technical features of the system. So Vilber is a leading life science company whose headquarters and manufacturing plant are based near Paris in France, with regional offices in Germany and China. Vilber has a presence in over 100 countries through a network of established distributors, and we have equipped more than 20,000 laboratories around the world. A bit of history first. Vilber was created in 1954 and started as a small startup company manufacturing UV instruments such as UV cabinets, DNA crosslinkers, radiometers, as well as UV lamps. It's from the 1980s that Vilber started imaging systems with the release of gel documentation systems for DNA, RNA, and protein gel imaging, like the DocPrint CX3 that you can see there, for protein and DNA gel imaging. And 2002 was a transitional year for Vilber, who were the first manufacturer to equip imaging systems with a fixed focal length for chemiluminescence detection on Western blots, having the widest lens aperture to collect a maximum amount of light and reach unrivaled levels of sensitivity. In 2011, we started to receive some publications with images of small animals taken with our Western blot imagers like the Fusion FX7 that was providing already great sensitivity for in vivo imaging of bioluminescent and fluorescent signal. That is how the idea of a proper instrument dedicated to small animal imaging emerged, releasing the Newton 7 Mini in 2016, which would include animal management accessories like a heated bed and nose cones being compatible with any anesthesia stations. But we did not want to stop there, as we wanted to provide our customers with a system that would push the limits of 2D imaging, featuring the possibility to better understand deep tissue structures. Thus, the launch of our brand new Newton 7 FT, that is integrating 3D optical tomography on top of bioluminescence and fluorescence applications. So Vilber is proud to release its brand new optical imaging system for in vivo, in vitro, and ex vivo imaging applications. The Newton 7 FT is dedicated to bioluminescence detection as well as fluorescence detection, providing superior quantitative results with its unique 3D tomography expertise. For non-invasive imaging of small animals, a variety of different modalities are available, from X-ray micrography, MRI, optical, PET, CT, or ultrasound. The Newton 7 is an optical imaging system, which is one of the most flexible and effective imaging modalities for preclinical animal studies. First of all, the Newton 7 is a user-friendly instrument. There is no need to be an imaging expert to master the system and all its features. As an optical imager, there is no radiation emitted from the Newton, which makes it easier to set up in the laboratory since there is no strict regulations like all the other modalities. It is possible to perform longitudinal studies in a non-invasive manner, reducing drastically the number of laboratory animals killed every year. And you can run large throughput studies with the simultaneous visualization of up to 5 animals. With the Newton 7, you can monitor the growth of tumors and infections, track cell migration, target molecules to nanoparticles, and visualize vasculature and microcirculation. It is compatible with any rodents such as mice, rats, guinea pigs, and small rabbits, zebra fishes, ex vivo organs and tissue sections, as well as in vitro samples in microplates or Petri dishes. So that was it for the quick introduction of Vilber and the Newton 7 FT. Now I'm gonna let my colleague Esmeralda going through some bioluminescence and fluorescence case studies.
Thank you, Alexis. As mentioned previously, our systems can be applied in various fields of research, both in bioluminescence and fluorescence. Our systems represent a powerful approach in numerous projects, research in the visualization and analysis of results. So application of bioluminescence imaging has grown tremendously in the past decade and has significantly contributed to the core conceptual advances in biomedical research. Bioluminescence, which is a naturally occurring phenomenon in organisms from different genera, is characterized by visible light emission in some living organisms. It comprises activity of an enzyme, the luciferase, that catalyze light-producing chemical reactions in these living organisms. Although many luminescent species exist in nature, 3 luciferases have been studied thoroughly and are used in biomedical research, namely the firefly, the CPNC, and the copepod. The table shows the different luciferases commonly used and the chemical reaction with their substrate. We can see that light is generated by a chemical reaction between the substrate, luciferin, oxygen, in which luciferase acts as the enzyme to catalyze the reaction. In a world where we are a lot more concerned about animal welfare in laboratories, bioluminescence is most commonly used for in vivo optical imaging. Today, molecular imaging offers many unique opportunities to study biological processes in intact organisms. This technology provides valuable means for the monitoring of different biological processes for oncology, immunology, nanomedicine, virology, and neurosciences. Bioluminescence imaging is simple to execute and enables monitoring throughout the course of disease. It allows for the non-invasive study of ongoing biological processes in small laboratory animals, allowing localization and serial quantification of biological processes without killing the experimental animal. This powerful technique can reduce the number of animals required for experimentation, since multiple measurements can be made in the same animal over time, thus minimizing the effects of biological variation. So we have chosen a few examples, proof of the consistency of the results obtained with our system. The first case study of bioluminescence imaging is in oncology. This study has been carried out by Kai Conley and his colleagues in 2017. So they have been working on the local regional therapy with alpha-emitting trastuzumab against peritoneal metastasis of human epidermal growth factor receptor 2 positive gastric cancer in mice. So the aim of this study was to determine in vivo anti-tumor activity of local regional astatine trastuzumab in the treatment of peritoneal metastasis of gastric cancer in mice. First of all, a few words about the treatment used, such as astatine, which is a radioisotope which emits highly cytotoxic alpha particles. Those alpha particles are one of the most potent cell-killing agents available, but which spares the surrounding normal tissue. The other treatment that has been used and tested is trastuzumab, which is a monoclonal antibody that has been used previously as a targeted therapy for human epidermal growth factor receptor, abbreviated as HER2. HER2 is a predictive biomarker of gastric cancer. Thus, trastuzumab induces an immune-mediated response that causes internalization and downregulation of HER2. So during this protocol, they designed it to be able to evaluate the therapeutic efficacy of astatine trastuzumab against HER2 positive PMGC in a mouse model. On the right is a schematic representation of the treatment schedule, as you can see. At first, the PMGC mouse models, as you can see over here, were established by IP injecting luciferase in transfected cells into 5-week-old mice 1 week before the experiment, as seen over here. On day 0, there has been an establishment check On day 1, the PMGC mice received a single IP injection of either PBS, unlabeled trastuzumab, astatine, astatine trastuzumab to 1 MBQ, or astatine trastuzumab at 0.1 MBQ. So on day 1, the PMGC mice received a single IP injection of those different treatments. 10 minutes after the injection of luciferin, bioluminescence from PMGC mice model was captured for 10 seconds. The tumor growth was monitored longitudinally in each group by our in vivo bioluminescence imaging. Images were captured on the day before the treatment, that is on day 0, and at 3, 5, and 7 weeks after the treatment. On the right are representative bioluminescence images of the tumor progression of the mice from each group. So we can observe the evolution of the signal with time and according to different treatments and different concentrations. The tumor growth was obvious in the PMGC mice injected with PBS, with the trastuzumab, the free trastuzumab, and the astatine, and at 0.1 MBQ astatine trastuzumab. Trastuzumab and PBS injections had little to no effect on tumor growth in the PMGC model. The tumor remained unaffected. The tumor growth appeared to be stable until 20 days after treatment with free astatine and resumed thereafter. In contrast, with astatine-labeled trastuzumab at 1 MBQ, no signal has been detected. Here, a single injection of 1 MBQ astatine trastuzumab reduced the tumor burden at 3 weeks after injection, with the tumors becoming undetectable at 5 and 7 weeks. Meaning that, as expected, it specifically targets and ablates HER2-positive human cancer cells in vivo, as seen from the image captured, thus successfully inhibiting tumor growth and improving survival in the PMGC model mice. They have been able to conclude of a downregulation of HER2 by astatine trastuzumab. In this first case study, the imaging performed with our system enlightens that local regionally administered astatine trastuzumab efficiently treats HER2-positive PMGC in a mouse model. The second case study of bioluminescence imaging that I will present concerns the infection field. Here is a study carried out by Rodier and his colleagues in 2017. They have been working on the in vivo bioluminescent tracking of enterotoxigenic E. coli mice. The aim of this study was to evaluate the colonization dynamics of enterotoxigenic E. coli, named as ETEC, by performing bioluminescent tracking during in vivo mouse infection. Here, in vivo tracking of bacteria allows to understand the role of ETEC during bacterial infection. ETEC being a leading etiologic agent of diarrhea worldwide, causing thousands of deaths every year. So in this first experiment, they generated a vector harboring the luciferase gene under the regulation of DNAC gene promoter, resulting in the PR-MKLUC vector. Then E. coli strains were cultured and incubated with luciferin for 1 hour and IP inoculated into 4 weeks old mice. They then IP inoculated the mice with either E. coli K12 harboring the vector or E. coli K12 harboring PBR, which is a dimer of E. coli. After 1 hour of infection, bioluminescent signal emission from the animals were captured. Bioluminescent signal is depicted on the right as pseudocolors, red being intense and blue less intense. The arrow indicates the bioluminescent signals. So here again, Vilber system allowed the tracking of bacteria during passage through the mouse intestine. In this first acquisition, consists of a control experiment showing that mice infected with E. coli K12 harboring PBR322 did not exhibit bioluminescent emission. However, mice infected with E. coli harboring the vector emitted signals in the mouse inoculation zone, as shown on the image here. In a second experiment, ETEC strains were electroporated with the generated vector. Here, the streptomycin treated bulb mice were IP administered with luciferin and gastrically gavaged with ETEC harboring the vector and E. coli harboring also the vector. Following gastric inoculation, mice were anesthetized and immobilized to capture bioluminescent signals. Light capture began following gastric inoculation. Bacterial passage through the mouse intestine determines the colonization dynamics of intestinal pathogens such as ETEC. As a result, at 0 hour, bioluminescent signals in mice inoculated with ETEC harboring the vector indicated bacterial passage through the mouse intestine. At 48 hours, gastric inoculation indicated bacterial passage through the mouse intestine. Over here, 120 hours post-inoculation, bacterial bioluminescent signals remain in the mouse intestine. So E. coli K12 bioluminescent signals were localized in the cecum. After 120 hours of E. coli infection, the mouse gastrointestinal tracts were extracted to perform ex vivo imaging. The intestinal tract dissection comprised the esophagus to the rectum. Here, a bioluminescence ex vivo imaging of bacterial passage through the mouse gastrointestinal tract is shown on the right. Ex vivo imaging allowed us to locate the region of the intestine colonized by ETEC. We can clearly observe the progression from 0 to 48 hours and 220 hours. The bioluminescent signals emitted by ETEC harboring PRM-KLOC were located in the proximal mouse ileum, approximately 6 centimeters from the cecum, whereas the control signals were identified in the cecum and in the proximal colon, as seen on the figures. To conclude, it has been clearly demonstrated here again that our system has shown that ETEC colonizes the mouse intestine and exhibits tropism for the mouse ileum. It has also be able to see the easy visualization quantitation of host pathogen interactions in live animals. Furthermore, our bioluminescent images permit the extent of pathogenic infection to be determined in real time in living organisms, providing temporal and spatial information regarding labeled bacteria and their metabolic activities. Now let's have a look at the possible applications in fluorescence. First of all, a little recap on the fluorescence imaging principles. Fluorescence is the phenomenon during which certain molecules under illumination absorb light of a specific wavelength and normally react by emitting light of a comparatively longer wavelength. This phenomenon relies on the production of excited single states. An electron in the excited orbital is paired with the second electron of opposite spin in the ground state orbital. A photon is emitted as a result, causing a rapid return to the ground state. We also talk about wavelengths, which are known to determine the degree of absorption and scattering in tissue, and therefore the penetration depth in tissues. Using near-infrared light permits detecting tissue structures deeper under the surface than using visible light. In particular, near wavelengths were observed to travel more efficiently through tissues than ones in the visible light spectrum. The fluorescence can be applied in numerous fields of research, one of which is of particular attention today is in nanotechnology. Nanoparticles have emerged as attractive drug carriers for intracellular delivery in the field of nanomedicine. To achieve an optimum anti-tumor efficacy, those types of nanoparticles have been developed to enhance the intracellular delivery of therapeutic molecules. Here, our customer from Nanobiotix proposes a new approach to change the way nanomedicines are biodistributed by priming the body to receive the treatment. The benefit of a therapeutic agent comes from its bioavailability, its intrinsic efficacy, balanced with its toxicity profile, as shown over here. So let's have a look at this little video.
For most drugs, treatment outcome is limited by drug distribution in the body. After administration, a drug enters the general blood circulation, then passes through healthy organs and tissues, some of which are involved in drug clearance. This means that not all of the drug will be useful. The liver, an important organ for drug clearance, contains hepatocytes and Kupffer cells, which trap and eliminate a wide variety of compounds, including drugs, according to their physicochemical properties. After hepatic clearance, only a small portion of the initial dose remains available in the bloodstream to accumulate in the target tissue. This poor bioavailability may lead to lack of drug efficacy or adverse events. How can the benefit-risk ratio of drugs be redefined? The nanopriming technology platform allowed us to create nano-sized objects called nanoprimers that could prime the body for treatment. Nanoprimers have been designed with optimum physicochemical properties that allow them to transiently occupy specific liver cells. Nanoprimers are made to be injected first to go in the liver cells. Next, the drug is injected into the body. Since the liver cells are busy, more of the drug remains in circulation. As a result, a greater proportion of the initial drug dose is available for potential accumulation in the target tissue for therapeutic action. The nanopriming technology platform could benefit current treatments and optimize the bioavailability of many existing drugs. The nanopriming technology platform may benefit a wide variety of existing products and inspire new approaches to drug design by redefining the relationship between bioavailability, toxicity, and efficacy. And it is brought to you by Nanobiotics.
Now let's go back to another case, which concerns the use of fluorescence in nanomedicines. This study was carried out by Nanobiotics in 2018. Here, the aim was to propose a new approach to change the way nanomedicines are biodistributed by priming the body to receive the treatment. In this design, so they designed a protocol to redefine nanomedicines bioavailability. To do so, they designed a liposomal nanoprimer with specific physicochemical attributes. This nanoprimer solution was then injected in the tail vein of mice. Then fluorescence acquisition were performed on the whole mice during 1 hour using our Newton. As a result, within 10 minutes following injections, most of the product has accumulated in the liver, with only a small fraction remaining in the blood. As seen on the picture over here, so there have been acquisition made with time at 5, 10, 20, 30 minutes, and 1 hour. So you can see that the nanoprimer has a specific accumulation in the liver after the intravenous injection. In a second experiment, they designed the protocol so as to determine if the nanoprimer increases nanoparticles' systemic bioavailability, and thus the impact of the nanoprimer on nanomedicine model bioavailability. Here, they use a PEG-coated polystyrene nanoparticles as a nanomedicine model. Fluorescent PEG-coated nanoparticles were intravenously injected in mice alone, or 10 minutes after intravenous injection of the nanoprimer. Immediately after the nanoparticles injection, the body fluorescence acquisition were performed on mice for 1 hour. As a result, an increased bioavailability of PS nanoparticles is observed for at least 1 hour when administered in the presence of the nanoprimer. So as seen on the right, so here are the injection done only with the fluorescent nanoparticles, and here it comprises the nanoprimer 10 minutes before the acquisition. So you can see the impact of the nanoprimer on the nanomedicine model bioavailability, and you can see that there is a specific marking. So here again, the imaging performed with the Newton enlightens the ability of the nanoprimer to increase nanoparticles, blood circulation, and their distribution pattern. In another case study, again using fluorescence in nanomedicine. So this is a study carried out by Sun and his colleagues in 2019. It was designed to test a combination of treatments, giving way to photodynamic therapy, which is a reliable and non-invasive therapeutic strategy for adjuvant treatment of cancer or other diseases. Here, the nanoparticles developed to enhance the intracellular delivery of therapeutic molecules are bismuth chalcogenides nanomaterials, which possess great potential in biomedical applications attributed to the high photoelectric absorption coefficient of bismuth and the anti-tumor activity of selenium element. Moreover, to be effective, these have to be coupled with other functional modalities for cancer theranostics, such as chlorine CE6, which is a photodynamic therapy agent with near-infrared fluorescence emission and high singlet oxygen quantum yield. So here, in this protocol, they developed a multifunctional nanoagent platform where C6 and doxorubicin, as a typical chemotherapeutic drug, were encapsulated into the tiny pores of bismuth selenides to form BPDC and CS, as seen over here. Moreover, PEG coated as the capping agent was conjugated onto the surface of bismuth selenide to enhance the biocompatibility and physiological stability, as well as stabilizing the encapsulated functional molecules. For fluorescence imaging in vivo, the mice bearing 41 tumors were intravenously injected with the BPDC and CS. The mice were then sacrificed at 24 hours post-injection, and tumors, as well as major organs, were excised for ex vivo imaging. As a result, at 24-hour post-injection, you can see the accumulation and enrichment of BPDC and CS in solid tumor region. So here are the different acquisitions that have been made in this experiment. So the ex vivo imaging also confirmed the higher tumor uptake and retention of the BPDC because of the prolonged blood circulation. So in another case study, which is actually the last one that I will be talking about today, it concerns the transport mechanism of doxorubicin-loaded chitosan-based nanogels across intestinal epithelium. This study, carried out by Feng and his colleagues in 2013, was designed to investigate the mechanism regarding the transport of chitosan carboxymethyl chitosan nanogels throughout the intestinal barrier, particularly focused on the cellular and molecular mechanism. This is another example of the use of nanomedicines in oncology. First of all, a little background information. Anti-cancer drugs administered through IV injection or infusion during chemotherapy entails numerous side effects due to the direct delivery of high concentration anti-cancer drugs to bloodstream. These inconveniences compromise the clinical treatment efficacy. Unfortunately, most cancer drugs, especially those with excellent anti-cancer effects, such as doxorubicin and taxanes, are not orally bioavailable owing to inadequate transport throughout the intestinal epithelium. So previous studies have shown that chitosan carboxymethyl chitosan nanogels could enhance the oral bioavailability of doxorubicin. In this protocol, cyanine 3-labeled chitosan and fluorescein fit has been synthesized, as seen over here. The biological material here that was used was overnight fasted SD rats, which were orally administered the doxycytosine complex to investigate the effect of calcium on nanogels-induced absorption enhancement. The acquisition was done at specific time intervals at 0 hour, 2, 4, and 8 hours. As a result, no apparent fluorescent signal were observed in the doxorubicin chitosan with calcium group during 8 hours detection. This may be due to the weakened capacity to binding calcium and doxorubicin being followed by a reduction in absorption dox. So let's go back to the image where you can definitely see the difference between the complex bearing the calcium and the complex without the calcium. So over here, the major organs were dissected at 10 hours after oral administration and observed by ex vivo imaging. And here again, the fluorescent signal with the complex bearing the calcium group detected only in the ileum and stomach. But for doxorubicin without calcium group, the fluorescent signal was apparently rich in the liver and the kidneys and the stomach, as seen on the diagrams. Well, before we move on, we're going to run another audience poll with Martin. Thank you.
Great. Thank you so much, Esmeralda. I really appreciate that. That was a really informative set of case studies, and we do have some questions that have come in as a result. We'll get to those at the end of the session. Before I run the poll, also just a reminder to everyone, please do use the Q&A dialog box to ask any questions you may have, and we'll spend some time at the end of the session answering those for you. Okay, and our second poll is, we'd like to know which substrates you favor for your research. Bioluminescence, luciferase-based assays is option number 1. Visible fluorescence, so GFP, M-Cherry, RFP, for example. Near infrared fluorescence is the third option. And again, there's a fourth option not applicable for you in case you're here for scientific curiosity. Interestingly, as a result of our first poll, 92% of our audience does use optical imaging, and I can see from these responses that many of you use all of these different substrates. That's really great to see. Okay, I'm gonna close this poll. I'm gonna bring our speakers back online, and Alexi's gonna continue on talking about the 3D optical tomography and some technical details of the Vilber Newton 7. Okay, now Alexi, go ahead.
Yep, thank you, Esmeralda. Thank you, Martin. Okay, so in this section, I'm gonna introduce you to our brand new 3D optical tomography module, which is integrated in the Newton 7 FT. So 3D optical tomography has been a valuable optical molecular imaging technique to non-invasively depict the cellular and molecular processes in living animals with high sensitivity and specificity. Then it could be co-registered with a digital library of organs and bone structures, and therefore it provides even more precise information on the signal's localization and size within the animal. What are the challenges of 2D imaging and how 3D tomography overcomes these challenges? In a planar view, the camera collects the different signal intensities from the surface of the sample. There is a lack of depth perception in 2D images that lead to approximate results, which can be under or overestimated due to the scattering light in tissues. The collected data is then misinterpreted. Using 3D tomography, the signal is reconstructed into a volume and repositioned into the 3D representation of the mouse, giving clear details on the signal size and its localization within the animal body. Vilber has developed a unique, cost-effective, and highly accurate way of achieving 3D tomography data. With the help of multiple 3D and IR cameras, the topographic image of the animal is acquired and reconstructed into a volume. Then, the main CCD camera acquires multiple images of the signal using various emission filters from wavelength in the spectral emission of the luciferase or the fluorescent molecular reagents injected. Eventually, the signal is reconstructed with the use of an algorithm and repositioned within the topographic image of the animal. The 3D tomography can be visualized with a library of digital organs and bones in order to better understand the signal's localization within the animal. Whatever the mouse or rat's size and position, the digital organs and bone structures will automatically adapt to its topographic image. You can choose to add or hide individual organs and bones, and you also have access to the axial, sagittal, and coronal views. The data of the signal is then quantified into a volume that can be measured or compared against other models or with the same animal over time. Now, in the last part of this webinar, I'm going to introduce the key technical features of the Newton 7 FT, as well as the animal management and the software features. Vilber has developed the Dark 9 camera, which is the latest generation of scientific CCD cameras, providing unrivaled levels of sensitivity for bioluminescence and fluorescence applications. The Dark 9 camera is a scientific 16-bit CCD camera, generating up to 65,000 gray levels with a dynamic range of 4.8 orders of magnitude, meaning that the very faint signals can be detected without saturating the strongest ones, ensuring a broad linearity with the capacity to quantify with confidence each and every signal within the image dynamics. The camera is grade 0, ensuring no dead pixels, and is calibrated to NIST standards. The instrument benefits from an exceptional image resolution with 4.6 megapixels. If the image resolution factor is not impacting the sensitivity, it still offers a high pixel density and thereby more details about the sample and the signal. High resolution is required in molecular imaging applications for better performance in pattern recognition and images analysis. It is then possible to generate high-resolution images with great sensitivity without the use of binning. Other systems with less than 1 megapixels may result in pixelized images. One of the important factors of the instrument is its lens aperture. The main function of a camera lens is to collect light. The lens aperture represents its capability to collect as much light as possible in a given period. Its sensitivity is usually expressed by a range of f-stops, and the smaller the f-stop number, the larger the aperture. A lower f number denotes a greater aperture opening, which allows more light to reach the CCD sensors. The aperture of the Newton's lens is f/.70, providing the best sensitivity and speed to reach the lowest limits of detection compared to other systems with smaller lens aperture. For bioluminescence, the time to get the image is dramatically reduced and precious substrate can be saved. For fluorescence, ultra-sensitive detection capability facilitates the use of shorter excitation exposure, thereby reducing photobleaching and phototoxicity and lowering dye concentrations. The Newton includes ultra-low dispersion components to enhance the sensitivity and aspheric elements to deliver consistently sharp images. Another important factor to consider is the camera cooling. Cameras generate heat, and heat generates noise in the picture, affecting the faintest signals detection. In order to better distinguish signals from noise, the Dark 9 camera is cooled to -90°C via 4 stages peltier thermoelectric elements to ensure the best signal-to-noise ratio, including bismuth telluride materials for a more effective and faster cooling technology. When the Newton 7 is switched on, it takes less than 3 minutes for the camera to reach the -90°C delta, whereas other systems take up to 20 minutes to be ready. There are infinite imaging capabilities with the Newton. The CCD camera is motorized on the Z-axis to accommodate various fields of view according to the number of animals to be visualized. You can choose from 1, 2, 3, 4, and up to 5 mice or 3 rats to be imaged simultaneously. On its lowest position, the camera enters the macro imaging mode with a distance sample to camera of only 6 centimeters to focus on a specific region of interest, such as the brain, and gain in sensitivity. Since the closer the distance sample to camera, the more sensitivity we get. Using the macro imaging mode, it is possible to focus on a smaller group of cells, detect metastasis easily, discriminate closely spaced signals, track cells growth or migration at the organ level or at a specific anatomic location. The heated sample stage is also motorized on the X and Y axis, so that the subjects are always at the center of the field of view, whatever the camera's position. Controllable from the PC, easily move the stage and focus on a specific region of interest. Then, the excitation light panels are also motorized in the X-axis to provide a unique scanning technique so that the excitation light is uniform all over the sample surface, which provide a more accurate fluorescence quantification. Speaking of excitation sources, 8 channels are included in the Newton 7 in order to cover the complete spectrum from blue illumination with 420 nanometers to 800 nanometers in the near-infrared spectrum. The powerful light generated by the spectra panels is categorized as laser class 2, producing a monochromatic light of a very narrowband illumination, reducing the cross-stimulation and increasing the sensitivity of your images. 8 narrowband pass filters are also included in the system, which are dual magnetron sputter-coated filter technology in order to ensure a transmission above 90% and a very narrowband cutting for improved spectral separation and increased sensitivity. Having multiple excitation channels in the system, you can then perform multispectral imaging. So it's possible either in bioluminescence or fluorescence detection in order to detect more than one target at a time within the same animal. Dual color bioluminescence can be performed using luciferase substrates of different natures like red firefly luciferase and green click beetle luciferase. Using multispectral imaging in fluorescence, excitations and emission spectra of the fluorescent molecular reagents have to be taken into consideration to avoid crosstalk issues. It is recommended to use fluorescent dyes with at least 100 nanometers excitation gap between the two. Here we have, for instance, the green signal acquired using M-Cherry, imaged with the 580 nanometers channel, whereas the IR dye 800 was acquired using the near-infrared channel at 780 nanometers. On the right, you can see the table of some of the most commonly used fluorescent dyes in preclinical imaging. So if you want to do multiplexing of multiple reporters, you have to choose them carefully to avoid the overlapping of wavelength. Crosstalk issues can then be manageable within the software to unmix the different wavelengths and reconstruct each signal individually. Talking of the software, Vilber provides a comprehensive software with various acquisition modes, either in automatic, manual, or serial, with the possibility to edit the images and quantify the results. You can find automatic image overlay for bioluminescence or fluorescence imaging. So the instrument will acquire the photographic image as well as the signal image consecutively in order to generate an overlay image, so the source of the signal can be localized with precision on the surface of the sample. Images that were acquired during different sessions can be grouped together to form a sequence. For example, a time series could be constructed from images acquired on different days following an experimental treatment. The software allows to collect and compare the data throughout the experimental treatment. You also have access to a movie mode where during the serial acquisition, we are able to accumulate all the images together to define a movie. So here, for instance, we have 60 images combined together to create this little movie. So you can see the biodistribution of the drug, for instance, or study kinetics. We have a unique 3D dynamic scan that images your sample in real time and reconstructs the different light intensities into 3D quantitative results. You can use the 3D dynamic scan with microplates, for instance, with different concentration of cells, and you can visualize them into 3D quantitative results. And eventually, the software also includes an intuitive quantification module where you can select various regions of interest to be compared to each other. The data is in counts per second, and you can also generate quick PDF reports or extract the data to an Excel table in one click. In terms of animal management, for the best ease of use and comfort, we can offer some anesthesia station or an isolation chamber. The anesthesia station is called the biosthesia, and it's made of a digital oxygen flow meter connected to an isoflurane vaporizer, in which the blended mixture of oxygen and isoflurane will create the anesthetic gas. We can use the injection box to deliver and make the initial anesthesia, and once the animals are asleep, they can be positioned within the dark room to be imaged and carried under anesthesia. The isolation chamber you can see on the right is to be used with immunocompromised animals. So this isolation chamber is sealed with HEPA filters so that pathogens cannot get in or out the chamber. So that avoids the risk of contamination. So we are approaching the 60 minutes. So I would like to thank you all to join this webinar, and also thank Cintiqa Instrumentation for hosting this webinar. And my last slide will be about a list of publications. So you can access this list of publications on Cintiqa's website if you want to have a look at them all. And Martin will run a final poll before we move on to the Q&A session. So thank you all so much again, everyone.
Great. Thank you so much, Alexi and Esmeralda again. We are sort of approaching the end of the hour, and we want to make sure we have time to get to some of the questions that have come in. And reminder, please, it's not too late to submit some questions if you have any. The final question, we'd like to know if you might be interested in a demonstration of the Newton 7 technology that we have presented today. We do often organize on-site demonstrations. So if so, which of these modalities might you be interested in? 2D bioluminescence, 2D fluorescence, or the 3D optical tomography feature that Alexi talked about? And again, this is multiple choice, so feel free to select more than one. Okay, while you are casting your votes, we'll just browse through some of these questions. Thank you everyone for submitting your answers. Okay, so Alexi and Esmeralda, I'll let you decide who answers these questions, but the first one is, what's the minimum number of cells that can be detected with the Newton system?
Okay, I think I will take this one. So the minimum number of cells that can be detected with the instruments can vary a lot of depending on the application and the animal model and the substrate that is used. But with the Newton 7, we had some end users that used the instrument to visualize, for instance, down to 10,000 tumoral cells. That was in bioluminescence detection. So we are talking about luciferase substrate that were detected into the brain of the animal. So it really differs from an experiment to another, where the bias factors are taken into consideration to image a minimum number of cells.
Okay, perfect. And I guess potentially related, another specification question, what's the maximum depth of light penetration for the system?
Yes, that's a good one, and it's pretty much the same answer, I would say, as it's also results of different factors, again, on the animal model, whether the mouse is nude or is very hairy, or it has to be shaved, and the substrate used also. So in bioluminescence, as long as there is a consistent signal, there should be no limitation. We could detect any cells emitting bioluminescence. In fluorescence, this differs a little bit. So we talked about the different wavelengths that can affect the light penetration within the tissues. So as we said, the visible fluorescence is usually difficult for the light to penetrate the animal. So visible fluorescence in the RGB spectrum could detect subcutaneous targets or on the surface of the sample, but they are not recommended for the detection of organs deep inside the animal. So for that, the near-infrared spectrum helps to travel better the tissue structures and detect targets to several centimeters, I would say up to 2 centimeters.
Okay, so the near-infrared imaging is the deeper, is the option to go with if you want to go deeper into the tissue structure.
That's right.
Okay. Do you generally recommend that the animal is shaved or that nude mice are used?
Yes, because the fur of the animal could alter the signal's position as well and the signal's detection. So even for near-infrared light, it is recommended to either use a nude mouse or to shave it entirely so that there is no autofluorescence at all coming from the animal.
Okay, perfect. You've touched on this before, but can you just maybe answer this again? Is it possible to image animals labeled with multiple reporters?
Indeed, yes. As shown in the presentation today, it is possible to label an animal with multiple reporters of different wavelengths so that the instrument will automatically scan the mouse with the 2 different excitation channels or 3 different. We can go up to 4 different reporters in one animal. So you just have to take into consideration the nature of your fluorophore. So they have to be wide enough from each to the other so that there is no crosstalk issues, so that the wavelengths does not overlap.
Okay. Perfect. I'm just keeping an eye on the time, and I'm cognizant of the time. I want to be respectful and make sure that people are able to get on with their next appointment. So let's maybe answer, 1 or 2 more questions. Very quickly, you mentioned an anesthesia system. If somebody has their own anesthesia system, is that compatible with the Newton system?
Yes, absolutely. So the biosthesia station designed by Vilber is an optional item, as we understand that end users may already have their own one. So it is totally compatible with the Newton 7 that welcomes a standard tubings to welcome your own anesthesia station.
Okay, great. Alexi and Esmeralda, you have access as well to the questions that have come in. Are there any there that you feel should be answered now? Again, we're not going to get to these all. We'll make sure to answer everyone's questions after the fact, but are there any on that list that you want to tackle before we sign off?
Yes, so let me have a look quickly. I can see, for instance, from Thiago Cardoso, is there any signal loss for imaging the brain through the skull? Is bioluminescence or fluorescence more sensitive for that region? So we imaged schools previously in well, brains, I would say, using both luciferase tumoral cells, which were providing great sensitivity. So I would say that the luciferase can go through the skull quite easily, so that would be okay. And we also tried with GFP during a demonstration, and that was not providing good results because, as said previously, the GFP is in the visible spectrum, and the lights was not going through the skull, unfortunately. So perhaps we could try with near infrared for fluorescence imaging, but this would be to confirm.
Okay, perfect. Well, I tell you what, and also there are some questions coming in to me directly through the chat window. So to all of you who have asked questions, Nicole, Ken, Marco, Suleiman, and Suming, we will address your questions after the fact. It is just after the hour. So with that said, I'm going to close off the webinar, and just we will also distribute a written transcript of all the questions that have come in and all the answers to those questions. So Alexi and Esmeralda, thank you both very much for your really amazing presentation today. It was very interesting, very informative. I think given the interaction with people and their engagement with our polls, I'm safe to say that everyone found it informative. And feel free to reach out to us at Syntica directly. Our contact information is on this slide. If you have any questions about the Vilber system, for those of you who have requested a demonstration, we will contact you to make those arrangements. Our next webinar is scheduled for January 28th and will feature Dr. Jeanette Woolard from Nottingham University as the guest speaker. And her talk is going to be very interesting and will highlight the use of Doppler flow technology to investigate hemodynamic effects of anti-cancer therapy. So hopefully you'll tune in then. Thanks again to all of you for taking the time out of your schedule to attend our session today, and we look forward to seeing you at another Syntica instrumentation event in the near future. Have a great day. And Alexi and Esmeralda, thank you again so much.
Thank you, Martin.
Thanks, Martin.
Okay, thank you all. Bye-bye.
Bye.